Thank you very much. I can promise I will be on time because I have a taxi at 4:45, so I'm leaving. But I'm hoping to capture an awful lot of information in a very short time. I've got a lot of slides, and I'm going to take you through classical biological control of weeds. We touched a little bit on biocontrol earlier, but I can tell you how it really happens. Everyone asks what CABI is, so we've been around 100 years, we're not for profit, we normally work in international development for developing countries dealing with their problems in agriculture and the environment. So our weed biocontrol work of around 30 projects a year is a relatively small part of CABI's activities. CABI is a global organisation working on behalf of 48 member countries, and we have a global activity and global presence, so we're quite active on the ground. And you probably know us best through the Invasive Species Compendium, and I'm going to be approaching some of you to write some new data sheets. Morpha is one we're commissioning at the moment, so they're written by experts and it's a very good resource. In Europe, we have about 36 scientists in 3 centres, all working on invasive species. We have quarantine facilities that enable us to work on exotic natural enemies safely, students and everything else. So we run about 30 projects a year on invasive species, invasive weeds mainly. And we've been around a long time and doing this a long time. So the basic fact on invasive species is they have an unfair advantage. They arrive without their natural enemies. The one thing that is always common in invasive species is they've lost their natural enemies. All the other traits are debatable, but they never have all their natural enemies, and that's always the true. It's not always the answer, but it's always true. Some of these species may be suitable for biological control if they are safe enough, and our job is to try and find the safe ones. Sometimes there aren't any. Heraclea mantegasianum, there aren't any. We've looked. Other species, there certainly are. And the unfair advantage is presented through performance. So here is Ulex europaeus, gorse in New Zealand. Gorse does not look like that in Europe, does it? So that's what happens when you take the natural enemies away. And if you spray with insecticide and fungicide, you can recreate this sort of performance in Europe by taking the natural enemies away and giving it the advantage. So this is a biological reason. There's 2 basic categories of weed biocontrol. Inundative, which I'll mention immediately, mycoherbicide using a fungus normally, native fungus, mass-produced, like the verticillium you spoke about earlier with Ailanthus, and applying it inundatively. And then there's classical, which I'll speak about, which is the main subject of my talk. Inundative biological control is normally used for high-value agriculture, things that are so valuable they can afford to invest in producing a product based on a fungus. Or perhaps where conflicts of interest exist, things that people like but you want to manage, but you don't want to use classical biocontrol, which will attack everything. So we work on things, well, better described as commercial. There's lots of scientific arguments about the names. As far as I'm concerned, it's commercial because it's a product that goes in a bottle and is sprayed, and someone pays money for it. So that's, to me, a very clear distinction about what biological control, inundated biological control is. And here's the kind of target we might consider as invasive. Not the Tesco trolleys, they are certainly invasive, but we're talking about Buddleia davidi from China. In New Zealand, they've released a clear pine weevil against this species very successfully. So there's successful biocontrol going on in New Zealand. UK, they love gardening. So nobody will accept me releasing a clear pine weevil to eat their butterfly bush in their garden. So that's a target you wouldn't do classical biological control, but you may consider inundative chemical analog biocontrol using a fungus. Here's an intermediate example, a very strange one, Stenopelmus rufinaceus against Azolla folliculoides. Almost all of you will have Azolla folliculoides as an invasive in your country, and you may notice that it just disappears with no reason. And everyone said it was a problem, and then it went red, not red, brown, and then disappeared. It's always to do with this weevil, I promise you. Look closely at any Azolla that's going a funny colour, stick it in a pot, put a mesh over it, and watch the weevils come out. What we do is we produce these, we overwinter these weevils. So they're not, this is not a classical biocontrol activity. This is an accidental introduction, continually being introduced as we're still selling Azolla in many countries. This is a contaminant, but basically it's a remarkably successful biocontrol agent. Not successful because Azolla is still a problem, but I'll show you why it's successful in a moment. We sell the weevil for cost, basically, for overwintering it, and we ship them out to people, hoping they'll apply it before the Azolla becomes a problem and the fish are throwing themselves out of the water. Unfortunately, they don't tend to do that, and they wait until August, and then they say, oh, my pond is dying, and we send the weevil, and the results happen. What's remarkable about this, as a biocontrol activity, is that it eradicates its host. Biological control programs are not supposed to eradicate their hosts, because then they eradicate themselves, because they need their host to feed on, because they can't feed on anything else. In this case, it's a freaky example because it's a new association. They haven't evolved together, and they have this mutual destruction when they meet. It's a really weird story, and a unique one, but a nice baby step into biological control for most countries, because we know this is safe and we know it works. This is before June 2008. A month later, starting to go brown, looking a bit dodgy, and then just a few weeks later, nothing. There is no azolla left in that system whatsoever. There's little fragments of dead azolla with 50+ weevils waiting for something to grow, and then they die. It's rather sad, but it's very effective. So you normally see the opposite of this. That's the before picture, and the after was the one you saw a minute ago. So classical biocontrol, the single release of a co-evolved natural enemy that is specialized on that plant. That's the dream scenario. We go back to the area of origin and find that organism, screen it for safety, and then release it. Often one single release will control on a national scale or regional scale. This is biocontrol of everything, insects and everything, not just weeds, okay? 7,000, this is not a novel approach. We've been doing biocontrol globally for centuries, and successfully for centuries. When it comes to weeds, it's a slightly different story. It's all based on a scientific concept, the enemy release hypothesis, just like a crop. You've got a pest on a crop, the crop doesn't go very well, take the pest away, the crop grows well. Simple scenario. That's pretty much what Keenan Crawley in 2002 captured. He was my PhD supervisor. Very annoying, because we've been doing biocontrol for over 100 years based on the enemy release hypothesis. We didn't give it a nice name, but Mick is a very clever guy. This is what biological control does. We have our weed population, an unacceptable weed population at the top. We release our biocontrol agent. There's a crash, and then an acceptable, economic acceptable, environmental acceptable, whatever threshold you wish to set. I've never seen anyone actually set a threshold because they never know what the result's going to be. But this is the ultimate scenario, and that goes on forever. That's the solution. One release, one investment, control forever, with some fluctuations. Does it work? Well, here's Eichhornia crassipes. We heard about this yesterday. This is a good example of biocontrol with some data. Neocotyna eichhorniae, a well-known biocontrol agent used all over the world, very successful. And these are the data from Louisiana, showing exactly the graph I just showed you theoretically in real time. 118 million acres. This is the scale of invasion that they were suffering from. You would call this a transformed ecosystem irrecoverable. They released the weevil, and that was the result. Of course, this is my personal bugbear, you could have another approach, the non-destructive and safe, although I wouldn't suggest if you lived on the bank that's safe, control of water hyacinth in the Guadiana River, where they basically burned 23 million euros pulling it out physically. Now, what annoys me most about this project was when it was presented to me, the guy said, we dismissed biological control because it would be irresponsible. And I consider this as irresponsible. And they spent another 5 million euros since then because it came back. So why not perhaps consider a longer lasting solution where you spend a fraction of that budget on looking at biocontrol, so when it doesn't work, you've perhaps got an alternative. So look for a cold-tolerant Neocotyna weevil. Still a possibility. So next time someone comes to you wanting this much money, maybe you want to think about what you want to spend it on, perhaps. This is a classical biocontrol success story, which we always talk about. Apuntia, Cactoblastis larva moth against Apuntia in Australia. 50 million acres invaded. That's the whole size of Great Britain. One massive invasion, impenetrable, unable to muster cattle, human health issues, all the other stuff. Released the weevil, the moth, and 200, I'm sorry about changing the levels of measurements, but 250,000 kilometres squared cleared completely. So you would have said, we talked about novel ecosystems yesterday, you would have said that ecosystem was a transformed ecosystem, irrecoverable, we have to just live with it. You wouldn't put that on your list of invasive species for Europe, would you? You'd say, that's absolutely irrecoverable, unless you happen to have a biocontrol agent that works. So I don't agree with the novel ecosystem theory, because there's no way back from a major ecosystem change. And in this case, there is. Another example we were involved in, this is a climax ecosystem in Australia of eucalypt forest. Doesn't look like a climax that we know, but this is what they have as a climax ecosystem. Uninvadable. Climax ecosystems are uninvadable, aren't they, supposedly? Unless you climb over them. And here's rubber vine weed, Cryptostegia grandiflora rather. A disaster, like a green custard just poured over the plant. Nothing's recovering from this, nothing will survive, and this is a World Heritage Site. So this is the end of the future of that, until we found a rust fungus in Madagascar, which has now been released and is doing the same as the Cactoblastis moth. So we're having native asclepiads recovering, native grasses recovering, and a huge success story. So this is probably one of the biggest success stories in weed biocontrol. It doesn't always work, I must point that out, but I'll come to that in a minute. It's effective, it's been reviewed. The cost-benefit analysis are off the scale. If you ever ask an economist to consider whether it's worth investing in biological control, they will 100% say yes every time, because the small risk, the high risk of failure is offset by a very small investment compared to some of the money spent on weed management. It's safe, that's the most common question we get. Well, it's been used over a century, 400 different biocontrol agents, 1,300 introductions globally, and we end up with 8 examples of non-target effects. That's a pretty amazing performance. 7 of those 8 were predicted or are predictable by the studies, by the controls we put in place in the protocols now. And the worst ones were predicted at the time, and no one cared about the environment. They cared about agriculture, and they released things that weren't super specific. So the scientists did the work and said, this will attack that rare native species. But in 1972, who cared about the rare native species? So the science is sound. You can predict what it won't do. You can predict what the species will not attack. There is a relatively low success rate. So 1 in 3 is recorded as roughly the success rate for biological control. However, if you invest properly and you do it more sensibly, Australia quote an 80% success rate. If the saving in cost on control is more than the project cost, then that's a success as far as they're concerned, which I would agree with. So they have 80% success rate, so it's a fairly good investment. It was on the up, and now it's on the down. I'm hoping that Europe will be filling that back up again because we've done all the easy targets in 100 years, that's one reason. But also, the issue is Australia basically cut its budgets for everything, and they basically closed down biocontrol. They've just reinvested in it, thankfully, with $13 million. But also the US has been caught up in a legal claim about Tamarix and the white willow flycatcher. So that's blocked all biocontrol releases for the last 5 or 6 years. So that's why there's been a big crash in the latest bar. So I would say that biocontrol is probably on the wane because we've done an awful lot of targets and there's not a lot of investment going in, but we're just getting started in Europe and there's no shortage of targets for Europe. Historically, we've been a source of solutions around the world. Because of our history, we've been providing problems around the world by spreading weeds around the colonies and all the other things that we've been involved in. And we've very rarely benefited. And that's starting to change now as we've got involved in the UK. We wrote a paper 10 years ago with all the targets. Ailanthus should be on there. Yeah, there's a weevil for Ailanthus, for example, that the Americans are looking at, which looks pretty safe. So there's plenty of things. The red ones, no solution. The green ones we're working on at the moment. And the dark green ones are the ones that are successful at the moment. Unfortunately, most of our successes, or all of our successes in Europe, are accidental introductions. It's very irritating to me. Our intentional introductions aren't working yet, but the accidental ones are great. Maybe there's a lesson there, I don't know. Break the rules. So Japanese knotweed, I've spent my whole career, 22 years working on this species. Massive invader, this is the UK timeline. It's all over the UK. It's a huge issue for construction. It can devalue your home by 50%. It's just, you can't get a mortgage if Japanese knotweed is within 7 meters of your property boundary. A 100-meter garden, 7 meters away, you don't get a mortgage. So this is a major, major issue. It costs the UK economy £165 million a year, 200 million euros. So a very, very costly plant. A very good first target for biocontrol because everyone hates it. There are no knotweed fan clubs in the UK. So the newspapers love Japanese knotweed because of the word knot. You can play with the word knot in English a lot. So we knew there was a massive difference in natural enemies in the native range versus exotic range. Always the case for all invasive species. You go out and look at it, it's like concrete. There's nothing on it. In the native range, there's lots on it. We discovered 186 species of Phytophagous arthropods feeding on Japanese knotweed in Japan, plus about 42 pathogens. And our job is to screen those to find the 1 or 2 that may be specific. So most of them are dismissed. Only one root feeder, unbelievable, with a plant with such an enormous root thing with so many polyphenols in. You think that would select for some really cool specialist root feeders. None. We looked for 2 years to find root feeders, and we just found a big hepialid that we had to reject because it ate its way out of my luggage, as well as eating its way through anything else you put in front of it. So our job is to eliminate, through literature review, field observations, etc., to decide which ones are the specific ones. And you want to get as much of it done in the field because the host range testing is the expensive bit in a very expensive quarantine facility with expensive people like me. So you have to get the most data out so you minimize the time you have to spend in a rather dark room. So we rejected lots of beautiful charismatic species that I really enjoyed working with but didn't pass specificity. We don't want to release things that attack anything else because that's the end of my career, basically. So there's another safety valve. It's not just the science, it's my future. If I release something that attacks something important, I'm dead, basically, and as is the rest of CABI. So we're extremely careful. We ended up with Aphilara itadori. Itadori is Japanese knotweed in Japanese, so it's a pretty good start when the host is named after you. Does it work? Well, I can kill plants in the glass house easy, in the lab easy. So that's always the case for biocontrol. This is a time lapse with the psyllids on the left and not on the right, taken away and brought back every week to see what's going on. And over time, you can see the one without psyllids just keeps growing. The one with psyllids just doesn't grow. Every time the leaf emerges, the nymph is on it, it doesn't grow, it puts out another leaf, it moves away. So it's a very effective agent in the lab. And this is what I went out to the field with, this excitement about this potential agent. We went through a horribly complicated licensing process, which was the most complex in the world. I said that the UK was the most difficult place to release a biocontrol agent in the world at the time, because it was the first ever release for Europe. So we were being super, super careful. Public consultation, 3 months, 64 million people asked for their opinion, effectively, on whether we should be releasing it. Peer review, everything, and it ended up with the minister making a decision. So, but it set the scene. We were very, very careful, and we showed that it could be done in Europe. So the psyllid isn't working yet, much to my annoyance, but we now are able to release on rivers. We were restricted, we couldn't use rivers because we had to have a contingency plan. This was the first biocontrol program in history, weed biocontrol, with a contingency plan. Never in the world has ever had a safety net where you have to spray it and napalm it if anything goes wrong. So everywhere else in the world trusts the science. This was the first one in Europe. We've got to be super careful and keep the public happy. So we were very, very cautious. So we're now able to use riparian sites, which unsurprisingly is where knotweed lives, so we would like to think that's a more appropriate site with higher humidity. And we're also getting new stock from Japan, because the stock that we have has been through 130 generations in a Japanese summer in a laboratory without any winter. And then you're going to throw it out into Berkshire in the UK in spring. Not a good start to life after that long in the... So we've probably institutionalized these insects in the lab. They've been conditioned to lab environments rather than the field. So the fresher they are, the better. The Netherlands are very interested, so they've commissioned us to do a PRA. That PRA is now available. That's 100 and something pages of data if you want to, interested enough to read it. And we have a paper you can read as well. I could send that to you. And Canada, as of a week ago, are now releasing the psyllid in Canada. And they'll do it properly. They'll throw the kitchen sink at it, hundreds of sites rather than 8 sites, which is what we're doing. And the USA is at the bottleneck I mentioned before. They're stuck in the aphids are terrified of licensing anything at the moment. So I keep forgetting this is being videoed. I shouldn't be saying things like that. But okay, so there's a problem in the US with regulatory approval. Another spin-out of the project is that we discovered a potential mycoherbicide. So there's a thing called Mycosphaerella polygonicus, which causes this damage. This is field damage. This is not us in the lab. So this is a devastating pathogen all over Japan, and we've discovered that it has mating types. So if you release just one sex, if you will, then it can't ever establish and it can't infest the species that we had worries about in the host range testing. So this could be coming to a knapsack sprayer near you in a few years' time, 10 years' time, who knows. I don't know what the licensing process will be, but this has got great potential. A non-chemical solution to Japanese knotweed, hoo-ha. Himalayan balsam, horrible problem. And you've seen this. This is a river in Cornwall. We were able to go back to the native range in the Himalayan foothills, discovered various things. We ended up with a rust fungus. We actually had to rename it. It's new to science. We had to rename the rust fungus because it has a varietal difference. It doesn't attack parviflora. It does attack glandulifera. So we had to distinguish it from the parviflora rust, which we did. So this species is now accepted. This is the life cycle. The great thing is it's an auspicious rust, and it hits the same plant twice. It hits it as a seedling, then it goes through and sporulates as Uridinia, reinfects, and then goes on to ascospore. So this is a very neat cycle, hitting the plant twice. It takes out 90-something% of the seedlings in India. We've never seen an infected seedling survive to adult in India. And Puccinia komarovi has been effective against parviflora in Hungary and other countries. where it takes out 98% of the seedlings. We've got the Himalayan balsam version of that pathogen, we believe. So this is happening now. We released late in 2014. This is the kind of thing, it etiolates the seedling. It's a really neat thing. It throws it above the canopy so the spores can get onto the rest of the plants, we think. These are the Urodinia. So we went through the process. It was Standing Committee on Plant Health. We're able to pass it after we produced a PRA with Europe included, the whole of Europe, not just UK, which was our mistake. And then we got ministerial approval. So this is out there now, and what we discovered is actually infecting seedlings the year after. We know that it affected localized plants, and we know that it can now infect seedlings after a winter. So this is a good first step. This is the beginning, 1 year. You know, we're just beginning this project, but very encouraging results. It's in the field and it's establishing. And I'm just winding up now. Acacia longifolia, I hope you've come across the work by Elia Marchante and Coimbra in Portugal. They just released last year this Trichologaster wasp, and this will be effective. This is a very effective agent. It basically neutralizes the plant, no more seed production. So you have to account for the continual seed growth after the seed fall, but eventually this will be crippling this species, as it has in Australia, as it has in South Africa. So for the future, floating pennywort, this is a project we've been doing for about 5 or 6 years now. We found a weevil, Listerionotus elongatus. Weevils and floating weeds is a marriage in heaven. They hate each other. It works perfectly. So this is Listerionotus elongatus, and there's the larvae that mines the stem and the stolon, the petiole and the stolon. So this should be a very successful agent if it passed safety testing. It has some feeding on non-target species as an adult. 1 minute, I'm there. Crassula helmsii, another shocking plant. It seems to recognize Ramsar status and invade. And we've got an eriophyid mite, tiny. This is a tip of 1 bit of Crassula, and these are all the mites on the tip. It's an incredibly small species, but it's an eriophyid mite, and they're famously specific. So we're quite hopeful for that one. And then future targets, well, it's on the list. Ludwigia, what a dream target that would be. And the Argentine colleagues that we have been working on this for the US for some years, and there's off-the-shelf agents ready to go. We have a rust and a thrip species that we're petitioning. We're asking the French to invest in some research to show that this is specific, so watch this space. And then, of course, there's the elephant in the room, Ophrya comuna, hammering ambrosia in the field in Italy and Switzerland, to the point where airborne pollen recording is showing a massive crash in airborne pollen and the allergenic effects in the population and the environment. This is an unanticipated biocontrol success, and not the agent we would have released because it likes sunflower occasionally. So, but it's happening out there. So biocontrol can work. It's possible to do it. We've been an early adopter in the GB, but there's other countries involved now. We've released an insect, an exotic insect and a fungus. We hope to release a mite and a mycoherbicide. So everything is moving quite fast in Europe. It is a plant health issue. All regulations should be through plant health because these are organisms likely to injure plants in the field, whether they're exotic or not. So plant health is the right regulation, and it's not easy, is my only conclusion at the end. And lots of people involved in these sort of things, of course. So thank you, and sorry if I ran to time, over time.
Thank you, Richard, for the impressive presentation. Any question related to this issue? Can we have a microphone? There.
Thanks. Thank you for a very interesting presentation. I have a question, can be used for biological control alien enemies of this species? And can we introduce especially enemies for biocontrol? And if yes, is it not a source for, a reason source for invasion of enemies in this territory?
Yes, yes, you can. All those species were exotic. They're all non-native. So you can do it. It can certainly happen. The safety testing that we do makes sure that they won't attack anything else. So we know that they won't do that. Whether they will become invasive, yes, they're an exotic organism that will expand their range and invade, but they won't have any negative impacts. They will only negatively impact on the target species and the target species alone. Nature has selected them to be specialists, So they should not move on to anything else. They're under the same evolutionary pressure as every other species in the environment, including those that are threatened because their host is suffering because of man's intervention, and they aren't moving on to new species. They're dying with their host. So the sort of inbuilt DNA of the organism suggests they won't move very quickly. Eventually everything evolves, but it should be spayed. It's a very good track record.
Hello. I was wondering about the regulation, because I don't know if you mentioned if is it possible to introduce this species in all the countries in Europe, or it's just...
I see no reason. We've done it in Europe. UK is in Europe, although we pretend we're not.
Okay, but
we may
apart
not
from...
be. We might not be in a month's time, but...
Apart from the UK, I don't know, because I don't know this regulation, but in Spain...
It would be necessary in each country to carry out a pest risk, or to use the pest risk analysis and follow your own laws. And in continental Europe, it's rather more difficult because you should be discussing with your neighbours rather more carefully than perhaps the UK, because we have a, a break. So yeah, but of course, it's every country in Europe interprets the same regulation in a different way, but they all prevent the introduction of alien organisms, of threats to plants, and you just have to. Ours was the advisory committee on the environment. They decided. So the Wildlife and Countryside Act is the UK version, but Spain will have its own one, Portugal will have its own one, and it's been proven in Portugal. So yeah, of course it's possible. Begin with a Zola. That's always a good start. It's easy.
Last question, I would say.
Okay, thank you, Dick. As you showed, biocontrol can be very effective, but it won't eradicate the species. You will get to a new balance at lower numbers. Wouldn't the ideal strategy be then to use conventional methods like mechanical or maybe even chemical you understand, from the low numbers to actually get rid of both the target species and your biocontrol agent at once?
If your goal is eradication and you're obsessed with eradication, yeah. But if you're willing to accept an occasional presence of a species that exists in a native range quite happily in balance, then maybe you shouldn't try. And the risk if you do eradicate it and you eradicate your biocontrol agent, I've closed my quarantine facility and killed all the insects. They're out in the field now. You want to start again, it's a very expensive thing to begin again, because you'd have to screen it again and safety test it again, clear the pathogens out again. So keeping it in balance is a very good thing.